everybody. Welcome to Chapter 10. So we're going to be talking about biochemistry of our genes, basically, in this chapter. And so in the beginning, we're going to talk about some history about where we really discovered DNA and what DNA did. So the first individual to discover or identify DNA was Frederick Michener. And this was, when he discovered it, he actually called it nuclein. His grad assistant, Richard Altman, then coined the term nucleic acid when he recognized the acidic nature of the nucleon. And then Albrecht Kossel isolated and characterized the 5 different nucleotides that make up nucleic acid. So adenine, thymine, guanine, and cytosine, as well as uracil, which is found in the RNA. We all have heard of Gregor Mendel. Hopefully you've all heard of him, and hopefully you already know a little bit about Mendelian genetics. So Johann Gregor Mendel was the first individual to come up with a hypothesis on how heredity occurs, right? And he studied pea plants. So he presented his research in 1865, and he published it in 1866. So he worked on pea plants for years, and he was always using true breeding, which means homozygous pea plants, so organisms that have the same type of characteristics, so dominant, or recessive. And so he got these really easy numbers to follow, correct? Or understand, hopefully you understand that. He did publish his work, but he didn't get credit for it. He got credit for the work he did, but he didn't really get a lot of credit for, people didn't actually believe his work. They still had this idea that we were blended. So basically mom's characteristics and dad's characteristics blended together to produce their children, which is not what happens. And we know that now. But at this time, we didn't. It wasn't until Theodore Boveri and Walter Sutton, working separately, but in the same year, 1902, they rediscovered his work, and Theodore Boveri was looking at embryonic development in sea urchins, and he noticed that it was nuclear components that determined that development. Walter Sutton then identified the separate, or was able to separate chromosomes during meiosis. And so both of them helped in developing the chromosomal theory of inheritance, which was associated with Mendelian inheritance. Oops, sorry. Then Thomas Hunt Morgan in the mid-1900s, 1915, that's what I thought. In 1915, he carried out hundreds and hundreds, it might have even been thousands of fruit fly experiments. But he worked for years on, so it had to have been thousands, because if you're working for years, fruit flies, they grow, I mean, it literally from the time they come out of the pupa to the time they are sexually active is 8 hours. So they have a very, very short lifespan. So I'm sure he did thousands of experiments on fruit flies. Him and his associates then published a paper in 1915 called The Mechanism of Mendelian Heredity. So now Mendel was starting to get credit for all of his work, his previous work. Then in the 1920s, the late 1920s, BarbaraMcClintock developed a mechanism for staining chromosomes, so now we can look at chromosomes. She also identified certain chromosomes that seem to break apart and recombine in different areas, and she called them jumping genes. We now know they are transposons. So all this is happening, and we still don't actually know if DNA is genetic material. But right now we do have an idea that whatever the genetic material is, it's in the nucleus. And so ShemHammerling actually established that the genetic information was housed in the nucleus, and he did this. He was studying a certain type of fungus called acetabularia, and so that's what this is right here. And so he would take the cap off of the acetabularia, of some of the fungi, and then he would take the feet off of other fungi of the same genus, right? The nucleus was housed in the foot. So if he removed the foot, there was no nucleus. But if he removed the cap, there was still a nucleus. And if the cap was removed, the cap could regenerate. If the foot was removed, the foot did not regenerate. So this is how he established that the nucleus was the house of genetic information to allow for reproduction of cells. And then he went a step further, and he was looking at two different types of fungi in the same genus. So he looked at Acetabularia mediterranea and Acetabularia crenulata. And what he noticed, or what he did, was he took the foot of the Acetabularia mediterranea, and he would graft that onto the stalk of the Acetabularia crenulata, and vice versa. And so what happened was, if he grafted the foot, the Acetabularia crenulata, onto the stalk of the Mediterranea, the flower, or the cap, would become the cap of the same Acetabularia crenulata that the foot was. And if he grafted the Acetabularia mediterranea foot onto the stalk of the Acetabularia crenulata, then that one would produce the cap of the Acetabularia mediterranea. So now we know that the genetic information that's carried in the nucleus codes for what the cap looks like. While Yoshem, I think that was it, yeah, Yoshem was working on the Acetabularia, George Beadle and Edward Tatum were working on the red bread mold called Neurospora crassa. And what they were doing was they were irradiating the DNA within the Neurospora, and that caused mutations within, they called them mutations within the DNA sequence. Then they would grow the fertilized mold on different media. And this allowed them to see if they grew the mold on a specific type of media that didn't contain all of the nutrients, they could identify what type of mutation had occurred. So if a spore, in this case, cannot grow on the media that, like say it can't grow without arginine, then it has a mutation within the pathway of arginine, and so it has to have arginine in the media. Whereas another mutant might grow in a complex media, but not in a media that does not contain alanine. And so what they would do is they would then use, since they knew, okay, it's not growing in this media, or it's growing here, but it's not growing here, let's just add alanine and see what happens. Nothing happens. Threonine, nothing happens. Tyrosine, nothing happens. when they got to the arginine, they were like, oh, this bread mold has some mutation within the arginine production pathway. And in that, they were able to then grow the mold specifically with the arginine. And so this actually led to this one gene, one enzyme hypothesis. So now they were at this idea that one gene produces one enzyme. So if you kill off one enzyme, if you basically mutate this gene and this enzyme doesn't get produced, then you won't have the end product. And so they were able to do that using the irradiation and then the different media. So in the 1920s then, Frederick Griffith found that DNA, or hypothesized that DNA was the transforming principle. So he was studying streptococcus bacteria, and he had two different types of strep bacteria. He found that there was a smooth strain. The smooth strain had a capsule surrounding the strep bacteria. And then there was a rough strain of streptococcus bacteria. The rough strain did not have the capsule. And he was running tests. It was the same pathogen, same potential pathogen, Streptococcus pneumoniae. When he injected the mice with the rough living Streptococcus bacteria, the mice did not get ill. And so he said, okay, well, that's weird. But if he injected the mice with the smooth bacteria, living smooth bacteria, then the mice died. They became ill and they died. So then he was like, okay, let's do a heat-killed smooth strain. So he ran that test. And with the heat-killed smooth strain, nothing happened. So now he was like, okay, so rough living don't kill. Heat-killed smooth don't kill. So the capsule isn't the virulent portion. The bacteria aren't the virulent portion. So what's going on? But if you have the bacteria and the capsule together, then they kill. So then he looked at the rough strain that were living and the heat-killed smooth strain, both of the non-virulent experiments, and he put them together and the mice died. And not only did the mice die, but then when he looked in their blood, he was able to isolate the pathogen. But the pathogen was not just rough, there was also living smooth bacteria. And that he called was a transformation. So he said there's some transforming principle that is taking these rough bacteria and making smooth bacteria from them. So these dead cells are giving something to the living cells. And we know that the dead cells now are giving their DNA. So as they break down, their DNA floats out of the cell, and the rough cells can take that DNA in. And that principle is known as transformation, which we'll talk about a little later again. So in the 1940s, Avery, McCloud, and McCarty took Frederick Griffith's research, and they decided they were going to see what actually the transforming principle was. So they isolated the smooth Streptococcus pneumoniae. They then heat-killed the smooth bacteria and removed the bacteria from the solution. They then, in they then separated the solution out. So they had a control that just had the heat-killed capsules, and they then to that they added their rough cells. And what they found when they grew the bacteria on a media, they found smooth strain Streptococcus pneumoniae. They then did the same thing in a second experiment, but in this experiment they added proteases to break down any proteins that were present, and added the dead smooth bacteria and the rough cells, the living rough cells. And again, when they grew the cells, we had smooth Streptococcus pneumoniae. They did an RNase in an experiment, so they took the same material, added it to a solution with ribonucleases that would degrade RNA, and the result of that experiment was smooth cells. So you still, in each of these experiments, we end up with these smooth living Streptococcus pneumoniae. And we started with dead cells, dead smooth bacteria, and living rough bacteria. In one we added the proteases, in one we added the ribonucleases, in one we added nothing. Okay, all of them end with this living smooth strain. But in the last experiment, they added deoxyribonucleases. In the last experiment, they added deoxyribonucleases. So there was still protein and RNA in the solution. They added their rough strain of Streptococcus pneumoniae, and what they found was that there was no transformation. So you did not have those smooth living cells. The only time you have smooth living cells is if DNA is available to enter the rough cells. So DNA has to be the transforming principle. Hershey and Chase in the 1950s then confirmed these results. So what they did was they were looking at radioactive, or they were using bacteriophages, and they used bacteria. Bacteriophages are bacterial viruses. They used either radioactive phosphorus, which was labeled in the DNA, or they used radioactive sulfur, which was found in the protein capsid outside of the bacterial virus, okay? They then put the bacteriophage with the bacteria in a solution, let them hybridize. They allowed them to grow for a while, and then they basically centrifuged the cells so that the solution would be up top and all of the cells, the bacteria, would be at the bottom. So what they were looking for was to see where the radioactivity occurred. So what they were hoping for was that the cells inside, the bacterial cells, would have taken in whatever the transforming material was. So if it was the DNA, then they would take in that radioactive phosphorus. If it was the proteins, which a lot of scientists at the time still believed was the genetic information, was the transforming information, because proteins are large, then the bacteria would take in the protein capsid. And so they shook off, or they separated the viruses in solution from the cells. Oh, sorry. Oh, that's the last picture. I don't have another better picture. Well, okay, so I apologize for this. What I will say is, when they did this, when they looked for the radioactivity, when they used the radioactive protein capsid, so the sulfur, the radioactivity was in the solution up here, and the bacterial cells did not have any radioactivity. But when they used the radioactive DNA, the bacteria themselves contained the radioactive DNA, whereas the protein capsids were not radioactive, and so were not being seen. The radioactivity was in the bacteria. So this confirmed that the DNA, not the proteins, was the genetic information that was transforming these cells. I'm going to stop because this has been a long video, but hopefully you enjoyed the history. So I'll talk to you in the next video. Bye.